On June 2, 2012, DGP, the Center for Design and Geopolitics, held its second annual conference. Entitled Designing Geopolitics 2, it was held in the Black Box Theater of Cal IT 2, the California Institute for Telecommunications and Information Technology in La Jolla, on the campus of the University of California at San Diego. Now, we're going to end in a way near where we began, which was this correspondence between very large data, cloud platform, and specifically biology and biological data, biopolitical systems, biological metaphors, and these kinds of things, and the interweaving and interrelationship of these with digital systems, political systems, and the design problem that poses. The premise of taking synthetic biology seriously as a design medium begins with a prepositional projection of biotechnologies, including genetic, genomic, and transgenic engineering, on the design, architectural, industrial imagination, but leads us from there to several elsewheres. Such a proposition delinks conventional allegorical relationships between body and structure, and in their stead incorporates biologic and architectural bodies into indiscrete and reversible interiors and exteriors. Such a projection is populated by multiple virtual forms, cyborgs and transgenic bodies, generative tissue textiles, body architecture, hybrids, neo-Epicurean epistemologies, sexually replicating habitats, genetically engineered architectures, and building materials. The dark matter of recombinant design and its complex technologies of self are engendered by social, cultural, and ethical implications raised by using human, mammalian, or really any flesh as a design media, and they are enormously complex. This little organ design, flesh design, can be benign, such as Paul Laffoley's vegetable house, or horrific, such as the human skin lampshades of Buchenwald. But the risk of catastrophe won't likely dissuade global and local economies from amplifying genomic technologies and making real design interventions with them possible and perhaps desirable. But even as genomic and transgenic design destabilizes the very bodies with which we inhabit our shared worlds, what is most crucially at stake is not the integrity of the physical body per se, but of the local and global institutions built over centuries upon concepts of the body as a stable cultural referent, natural referent. At the same moments when the biological medium of the body fragments from a universal singularity to a genetic and digital assemblage, the worlds we define through the body become themselves equally destabilized and redetermined by design imaginaries. Any institution based on bodily discourses, which is to say any institution, is a potential site for some recombinant revolution, the family, the home, the nation state, and indeed space itself. And this leads us into several contradictory and sometimes dangerous directions. This is quite different, I think, from the postmodern multiplication of textual selves. It is a recognition that the only conditionally material membranes between body and the world are those that we design. The 21st century will be populated with genomically reflective and self-conscious children, born and growing in bodies known to them as inhabitable expressions of code. Are we populating the early years of a eugenic century with a nightmarish biotechnological singularization of humanity? Are we also participating in the first years of a new society of biomaterial freedom, an architecture of the self, of the multitude of communities that will allow and demand new reflexive practices of expression and purpose? Synthetic biology as a plastic design medium is Alexandra Daisy Ginsberg's trade. She's an artist, a designer, and a writer, interrogating science, technology, and new roles for design in a biotech future. As a design fellow on synthetic aesthetics at an NSF-funded project at Stanford and the University of Edinburgh, she's curating an international program researching synthetic biology, art, and design, investigating how we might design nature. Her works include The Synthetic Kingdom, a proposal for a new branch in the Tree of Life, chromi, a collaboration with James King and Cambridge University's Grand Prize-winning team at the 2009 International Genetically Engineered Machine Competition. In a short story written called The Well-Oiled Machine, co-written with Oren Katz while at Symbiotica in Australia. Recent talks include TED Global and PopTech. Her work was nominated for the Brit Insurance Designs of the Year 2011 Index Award, and she received the World Technology Design Award in 2011. So with this, we commence our headlining panel. Daisy Ginsberg, my pleasure. Thank you.
So, as Benjamin said, I'm a designer, an artist, and writer, an interdisciplinary sort of character, and it's really exciting to be here in this group today and found so many things that have already come up today that are in my talk. It's actually kind of really unusual that so many of these themes have already been covered. But what I want to talk about this afternoon is a new technology that's promising to change and even save the world, as all new technologies tend to do. And this technology is one that has never before been described as such: biology, by its very nature undesigned. And it's been coaxed by engineers into a design material. Ooh, sorry, I don't even know how to use a mouse. Where am I pointing? Sorry. Can we go back? Sorry, I think everyone may have guessed I'm a Luddite by now. Okay. So, this Hello World first statement of synthetic biology, 2006 iGEM entry from University of Texas at Austin. And it kind of represents a lot about synthetic biology, the boundaries of design extending into life, and the closing of a gap between what we make and who we are. Okay, I'm going to try and hit the right button now. Yes. So I wanted to add a subtitle to this talk because Benjamin said I was allowed to talk about the dark side of synthetic biology today. So I thought I'd add this, how I learned to stop worrying and love synthetic biology. And I am definitely a warrior still, and I wouldn't say I love synthetic biology, and I think that's precisely why I want to work in it. Because I think synthetic biology is defined by the complicated position that I find myself in as an outsider. This is a field where for some the desired endpoint is to save humanity from itself through the total industrialization of nature. That's actually a statement that I've heard said. And I think I describe this as the state of biological rapture. This thrilling Wonder Stories cover, and for those who are in this state of rapture, the only problem is the public's fear of biotechnology and technology in general. That's the obstacle, not whether the biology can keep up with the dream. And meanwhile, for the public, it's this image. Both women have this similarly rapturous face as they're engulfed by biology, but for the public, it's this biological dystopia, a future of ecological catastrophe. I think both are really flawed generalizations about where we are and where we're going. And I think the only thing that we know about synthetic biology is that if it does come true, its impact is going to be not just scientific and economic, but cultural, ethical, political, and absolutely above all, personal. And that's where I think design comes into it. Because we may impose human design on biology, but where do people enter this equation? We're saving humanity, but what about the individual humans? And that is the thing that I like to think that we should be investigating. If we're going to design nature, we need to understand what that means. So I'm going to talk a bit about synthetic biology and try and consider these people that the synthetic biologists are trying to save. And then propose some ways that we can begin to think about this and think differently about what's at stake. So synthetic biology, maybe most people in this room may or may not know what it is, but it's essentially a refreshed ideology for genetic engineering. Synthetic biologists identify genetic engineering as it has been as more like a craft rather than a proper engineering discipline. And to make biology into a material for the more efficient manufacture of systems and stuff not yet evolved by nature, we can just apply engineering principles. And this process of applying the engineering principles onto biology is a simple matter of the three tenets of standardization, the idea that a screw is a standardized part just as a piece of DNA can be a standardized part. The idea of abstraction, that you don't need to know what's happening under the hood to make it work. And decoupling, the idea that we can actually separate the biology and the design. And what this means within synthetic biology is this this is an upside down version of synthetic biology. It's a reverse hierarchy where DNA is spelt differently, but basically DNA can be abstracted into parts, and then into devices and then systems. And the idea behind this is that in the end, applications are all we need to concentrate on. That can be the design focus of this so-called biotech revolution. Everything else can just disappear under the hood. What this means is that machines are becoming biological and biology is becoming digital. BioCAD software suites, this is Clotho, which was developed at Berkeley, promise to take the design of circuits to the desktop. This idea that you can actually rationalize biology so deeply through human and computer logic can be rendered into a circuit diagram like this. And the idea that you can attempt to engineer digital behavior from structures that are fundamentally not digital is the absolute fundamental ideology of synthetic biology. To me, this is beginning after 5 years researching this field and looking at it from a design perspective. It's almost like it's an alternative branch of nature. I'll talk more about the synthetic kingdom, but it's almost the simplified nature. The digital biology is going to be something different. So this is Klebsiella oxytoca. Apologies if I've said it wrong. It's a bug that lives in your gut. And recently, Voigt and et al. Decided to understand one part of its genetic sequence, and that was the way that it fixes nitrogen. So this is the circuit that has been refactored. And what I find so interesting about this is this idea of refactoring. Instead of reverse engineering like you would an engine, you take it apart and put it back together again and work out how the thing works. This instead was taking synthetic genes and putting in between each identifiable part that existed in that sequence so that you could actually have a human logic part. This is that gene made simple. It's re-engineered and refactored through human understanding, but it only works at 7% efficiency of the existing sequence. And what this means is that the control is much greater, but the efficiency much less. And that's what I find really interesting, that this new biology somehow functions differently. It's not the same as what's gone before. And so if we think about this kind of rendering into physical, what I think is so interesting, I don't know if anyone here has been following this new aesthetic debate which kind of erupted at South by Southwest earlier this year, trying to identify something that's coming out of the digital culture that we live in. And someone called James Bridle described this as the eruption of the digital into the physical. But what I find really interesting about this is if we think about it... Oh, no, I don't find that. No, sorry, all my the PowerPoint's gone. Funny. I know, I'll make the point. For synthetic biology, I think it's actually truly the digital culture being rendered. It's the eruption of the digital into the living. And that is what synthetic biology is promising to do. It's almost an alternative evolution. So where is this eruption going to happen? Well, the kinds of funding and investment that's going on is very much in the kind of big industry domains. The malaria precursor, artemisinin, funded by Gates money of $42 million. Michelin investing to make synthetic isoprene for tires. Exxon throwing in $600 million to make algae-based biofuels. We even have DARPA now investing in its Living Foundries project to make cellular factories, also greening war with explosives that are made through bugs. And the most interesting, I think, is NASA, the idea that we can actually terraform Mars and begin to colonize outer space, that it's our moral duty to go further, that we can actually feed our astronauts and colonize new planets through synthetic biology. But I think there's a fundamental design flaw in this process. And to me, it's this. We're growing sugarcane in Brazil, and we're making it into jet fuel. And this, to me, is a disruptive technology that's promising not to disrupt anything at all. And I find it quite disingenuous. So that, I think, is the design problem that needs to be solved. This is sugarcane, a physical, biological stuff that's being made into goop in another country, and it's instead rendered like this. We have these amazing cyborg bugs and DNA made out of money. It's something entirely different. And I think it's best summed up by, on a phone call I had recently with someone in the industry, he was saying, well, academics describe it as a revolution. Meanwhile, us in industry, we prefer to say it's an evolution, because that's better for policy. Meanwhile, one of my colleagues said, who's a synthetic biologist, he's like, maybe it's just a delusional cloud. And I think it's really important to keep this in mind, because I believe synthetic biology will exist. It just might not yet. But it's stuff like this that we need to be worried about. And I just found this quote last week in a report. Many synthetic biology practitioners may wonder what is so offensive and morally alarming in the creation of artificial biological systems in the laboratory. This was in an EU-US task force report in the back section on opportunities to engage with the public. And I find this incredibly disturbing, because I can't answer why I find it offensive, but it's my human prerogative to take, to object. And that's where I think that we really need to think about how this, no, the slides have not come up. So imagine the tree of life. Imagine a technology that works. This is the tree of life, and this is our position. It's somewhere down the left in the eukarya. And the tree of life, of course, is an artificial construct. It masks so much stuff that goes on in nature. It's a model that we use to to make sense of all this stuff. Things like reproduction, symbiosis, diversity, complexity, all covered up by our model. There. Sorry, I don't use a PC. So this proposal I made a few years ago was that we just need to add an extra branch to the tree of life. This is the engineering solution to a problem of classification for genetic engineering. We're just going to put all this new stuff in. It's sort of somewhere in nature, but it's slightly different. But I think what's become really interesting from being involved within the field is how we deal with this kind of stuff, the commercial aspects of designing life. We're going to be dealing with obsolescence, style, uniformity. These are all things which aren't natural traits. And in comparison, these are the things which the material is best at. So how are we going to actually deal with this? So I want to talk about just a few little projects in brief, just to take bits of them, just to show how some of these issues can be made manifest through design. So Benjamin described this Eat Chromi project. Was an iGEM competition entry a few years ago by the Cambridge University iGEM team, and they designed bacteria that could secrete different colours. So this is an E. coli, which we renamed E. chromi, secreting violacin pigment. It's incredible. It was designed by 19-year-olds. And it was one of a series of colours. And this thing won the competition. And part of what we did was to work with the team. James King, another designer, and I spent the summer working out how, as designers, we could be involved in this process and getting the team to think about much bigger impacts. And one of the things that came out of the workshops was the idea of disease monitoring. There was some discussion this morning of disease monitoring and how it's problematic at the moment. It's always too late. You don't actually know when something's wrong. When something doesn't feel quite right, it's when you go in. Is there a way to completely transform that industry? So we were working on timelines, and we imagined... Okay, I can describe this project. Imagine a Yakult yogurt that you would drink in the morning, and it's E. chromi branded, you drink it. It's full of these engineered bacteria. And inside them, they have, each one has a circuit that detects different kinds of diseases. The bug starts to colonize your gut. You have a large colony of them. And if they detect a precursor of a different disease, a chemical signal, they start to secrete this colored pigment, engineered by 20-year-olds back in 2009. The obvious way for this colored pigment to be visualized is obviously in an external output. So each morning you go to the toilet and you check your shit. This is the principle of E. chromi, and we made it into a physical suitcase. Look, and I wish you could see it. I think the image is on the other computer, but maybe we can get it later. So this idea that a physical living computer could be something so visceral that it could actually be your poo was something that's captured a lot of people's attention. And taking that into the field and saying, well, we need to get away from this idea of this digital aesthetic and actually start thinking about biology as biology has been incredibly powerful. But what really interests me about this is how it raises so many different issues about what is wrong with the way this technology could go. The idea of obsolescence. Would you have to regularly buy this drink because the things are designed to kill themselves after a certain number of generations? How would you design accuracy? What level of risk would you be willing to take if the you're drinking genetically engineered bacteria that detecting diseases in your gut. So this has been a problem for me throughout this project, that the suitcase of shit travels around and everyone's like, yeah, I want coloured poop, it's great. Every day would be, a day of discovery. But that idea of self-discovery, do we want to be so close to disease and to risk in our lives? And how do we actually begin to rely on biology and begin to see synthetic biology as part of us could completely change the boundaries of what we see acceptable, and also change the boundaries of where commercial life becomes part of us. It's really worth seeing. Sorry, I don't know how to use this. Okay, another excellent video is of a plant cell dividing. The Hasselhoff lab in Cambridge works with plant morphogenesis and the way that plants grow, the way cells divide. And I went up back in 2009. I was working with a fellow artist, Sasha Poflep, and we were looking at this idea, the idea that you ship stuff around the world and how completely crazy it is, and how something like synthetic biology, a new technology, could be forced through happenstance to save the world. You buy a sofa, it's been manufactured in 3 different places and shipped backwards and forwards. And could biology take over? So this project was about trying to find a common product from this future. We decided a herbicide sprayer would be a contemporary commodity that would represent how objects could be grown rather than made. The thing itself was made of 7 different components that would be grown, harvested, and assembled. So just to quickly walk through a few of them, which we had them rendered in the style of a botanical history drawing to show how the thing would become validated. In contrast with the chromi project, which we set at 2039 at a particular date in the future because we felt that was when it would become culturally acceptable. This instead was a much sort of more poetic future. So this is the gourd full of herbicide which you'd put on your arm and you could squeeze out the poison. It would then be attached onto this connector fruit, grown in a bit similar way to a walnut. Next would be the nozzles that go on the end of the tube related to the tomato. The long translucent tubes which would carry the poison to the nozzle are grown in this sort of strange, elongated kind of plant. And throughout this process, we were talking with Jim Hasselhoff, the plant scientist up in Cambridge, and ring him up and be like, Jim, we're doing this thing, what do we need to consider? And he's like, you're mad, but maybe, cell differentiation, gravity, and tried to give us some sort of pointers. In the end, this was the final product. It was a herbicide sprayer. But the main point about this, it was bringing diversity and softness into a realm that's previously dominated by hardness and industrial manufacturing standards. The idea that products could be grown where sold, and you wouldn't, factories would become farms. And the main point was that seeds become the mode of information distribution, that you don't need to sell products, you just sell information. Seeds are the only thing you need to transport because all the manufacturing instructions are encoded into their DNA. And of course, this already happens with genetic modification. And this is the issue where I think that social issues are going to be so important. The control of seeds is already such an enormous geopolitical issue that I think it's something that synthetic biology can't sidestep. Because if we do end up in a future that is as seductive as one that is entirely sustainable and green, then things like who controls data is going to be more important than ever before, because this is part of us. We are made of this data. So this is the question that is driving the work I've been doing, is how do we design for a world we want when that possible world doesn't yet exist? I'll talk a bit about the Synthetic Aesthetics project. You're only missing a logo in this slide, it's not very exciting. But this is a really unusual project that was funded by the NSF and the EPSRC, which is one of the UK science funding councils. And Drew Endy, who's one of the pioneers of the field, he is one of the people behind the BioBricks concept, the idea that you can make DNA into parts, and the iGEM competition, which is a machine to further this whole process, stood up at a funding sandpit where about $10 million of funding was awarded to 5 different synthetic biology grants over 5 days. And with Alistair Elphick and Jane Calvert, who's a social scientist, they decided that what they needed to do was to ask questions like this. Can you design nature? How do you design nature? And how do you design nature well? And these were questions that we've been developing. I was brought into the project to try and find a way to get 6 artisan designers and 6 synthetic biologists together to actually look at these questions. So this was not about public engagement. This was about actually understanding what is going on from a design perspective. So we brought 6 artisan designers, from bio-artists like Oren Katz to smell artists like Cecil Tolas, to architects David Benjamin, who teaches at Columbia, working with generative algorithms, with 6 scientists across the field who work in really different ways, from protocells, so the bottom-up approach, building life from scratch, to people working with plants. So someone from Jim Hasselhoff's lab. I'm going to talk about a few of the projects just to show what I think is so unique about this project has been finding a way. It was so complicated the way the project was set up to get an artist and designer into the lab for 2 weeks. That's been done. But getting the scientists into the artist and designer's studio for 2 weeks and then seeing what kinds of discussion. There was no brief. We just said talk. So they had a month together, and all of these pairs are continuing to collaborate 2 years later, and that's what's been so exciting. So David Benjamin, who's an architect working with Fernand Federici, and they were really interested in this. This is a xylem cell. So the water transport system in plants is made up of these cells, and they build the cellulose structure where water flows through. And they were wondering how this could actually be used to reimagine what the definition of a biological computer is. So at the moment, synthetic biology is so fetishistic about DNA, but could you actually imagine the entire cell as the biocomputer itself? So the way that these cells grow and find their space and compute the structure that they build, can you begin to extract the logic of that and actually use that usefully both in architecture and in synthetic biology? So collaborating with Hod Lipson, who's a computer scientist at Cornell, they found ways to actually extract that growth pattern from the cell. And in doing this, the thing that really interests me is that I think it's an amazing philosophical challenge to synthetic biology. This idea that it's about the logic of the cell rather than the code, about the form of the cell rather than the code. These are all things that are fundamental to challenge the very kind of the existing 10-year-old notions within synthetic biology. So they were doing things like 3D prints to begin to try and examine what the structure was, but it was always about not following the kind of typical biomimicry path of just scaling up a cell, have a building that looks like a cell, but actually using it as a computer system. David went on to do some work with Autodesk, actually trying to work out how you could build plugins for programs like Maya to actually begin to develop biomaterials. But again, for me, this is an issue of can the biology keep up with the vision? But together they're working to continue finding ways to work with a biological analogy and challenge it. Sharif Mansi is a protocell scientist at Trento in Italy, whose basically his work is to build life. But the problem with doing that is you have to work out when you've made it. So I think every protocell scientist needs some kind of philosopher in the lab. But working with Sasha Poflep, who I did the growth assembly project with, they got onto this question. Could inanimate machines only have been an interlude in history? And I think this is such an excellent question for synthetic biology. The fact that we've gone from the horse to the steam engine, which was defined by horsepower, back to something more akin to a horse is going to be powering us. So working with a fine artist and a synthetic biologist writing papers that are more sort of philosophical, part of the question when we're writing our book proposal about all of this is like, is this synthetic biology? But I think these are the fundamentally interesting questions because they're being thought up by synthetic biologists and kind of collaborators in the lab setting, and it's incredibly useful. This is another interesting project. We decided to bring a commercial entity in from the design side. How would that work within a project like this? And remember, it's not funded by art and design, it's funded by science money. So you might remember the first image, Hello World. This is the same technology repeated, but with the IDEO logo. And for me, I find this very strange. Like, it's quite wonderfully sort of disturbing that IDEO are getting involved, and it's kind of my fault. But what's been most interesting about this collaboration between Will Carey and Wendell Lim is that it's really been about challenging the structures and the processes of design and science. So Wendell, who runs a very big lab at UCSF, I've just done it again. Sorry, I don't know. Is there a way to reverse this? No, let's go on. Okay, Wendell, who runs a lab at UCSF. Sorry, I can't. It doesn't work. So. Could you go back one more, please? Thank you. Wendell was really interested in the idea design thinking process. And for a scientist, he'd watched videos about this. He's like, how can we bring this into our lab and the way we innovate with synthetic biology? And I was like, well, I know someone at IDEO, maybe we can work out how this might work. And that's what's been so interesting, is that the collaboration has worked both ways. And for the IDEO team, they've been able to create space within the commercial practice to actually do kind of totally strange academic research. And that's had to go up to the top level of IDEO to make that happen. And I think that's been the biggest fundamental sort of thing that's happened there for both teams, is that they've created space to bring totally new collaborators in unexpected ways and actually challenge the way that things are done. And I think that's going to lead on to a much bigger collaboration between both kinds of disciplines. Oren Katz, who's the bioartist who ate the first piece of victimless meat back in, I think, 2004, and inherently critical of synthetic biology, was persuaded to work with Hideo Iwasaki, who's a scientist but also an artist. And this was a really interesting collaboration because both are hybrid people in a way. Hideo invites artists into his lab and makes art of his own. Oren runs an art science collaborative laboratory at the University of Western Australia called Symbiotica. And for Oren, part of what was so difficult is, as an artist, he didn't want to make anything useful, because that would be entering into the contract of synthetic biology. How could you make something critical but still make it out of biological material? So Hideo works with algae, cyanobacteria, and working on their circadian rhythm, so controlling the way their time clocks. And Oren is very fascinated by these stromatolites, which are at Lake Clifton and around Australia. These are accretions of deposited, I think it's calcium carbonate, made by cyanobacteria, by algae, over thousands of years. So these are actually living rocks and incredibly sensitive. And this Lake Clifton is under threat from salination, and these things could die. It takes about 5,000 years to grow something that size. So I think what they're going to end up doing is growing the world's slowest growing sculpture using engineered cyanobacteria. And I think it's particularly poetic because it really highlights the idea of geological time versus biological time. The idea that we're entering into this age of, or we're just entered into the age of the Anthropocene, this new era defined by humans, the kind of layers of plastic building up in the Earth's crust, which making a new geological era is quite a big deal, apparently. So there's been lots of people voting whether this is going to happen. But the idea that synthetic biology could redefine the geological crusts, that it could actually be an era where there is entirely new stuff in the Earth. Kind of just makes such a nice comment about time, about our time on this planet, and where our meddling fits into the much bigger scale of things. And finally, Christina Agapakis, a biologist who was at Harvard, now at UCLA, working with Cecil Tollas, international smell provocateur, who's kind of notorious for saying that there's no such thing as a bad smell. And as I followed this lot around the world, I decided I disagreed, but that's not the case. So Cecil's work is very much about how we live in this pasteurized, sanitized world where smell is not allowed. And so they decided to make cheese. And the cheese was going to be a special kind of cheese because the human body, as you may know, is covered in bacteria. And we cannot live without these bacteria. 1 in 10. I think our own cells are outnumbered by bacteria 10 to 1, but we rely on them to process food, to send signals to our brain. And yet we live in this world where you watch one advert as bacteria killing stuff, and the next one is for Yakult. And it's a very confused message for a world that's about to be saved by synthetic biology. So cheese is a culture full of bacteria, and it has great sort of bodily connotations. So there's smells in particular cheeses which make them so distinctive because it's the same bacteria that are found on your feet that are in the cheese. I think Limburger is the particular victim of that one. So cheese makers, especially artisanal cheese makers, actually make their cheese like this. And I was incredibly surprised to find this out. When I was visiting at Harvard with Cecil and Christina, they found this scientist who's setting up a cheese lab, and she's actually trying to look at cheese as a model ecosystem. So we're used to the mouse or E. coli as a model organism, but actually cheese as a cultural a cultured object and that could actually be used and analysed and studied as a great model system. Because the cheesemaker here, the stuff on her skin is ending up in the cheese, and it actually becomes part of what that cheese is. If you made it in a different place or with different people, it will taste different. So body cheese became the focus of this project, and they collected swabs and they made cheese. And my armpit cheese apparently is very fine. It was passed around about 100 people at the Media Lab, and they flew it to Berlin and smelt by a cheese expert, and they thought it was very good. But what they're actually doing is analysing what's in the cheeses, the smells of the cheese, and with an aim eventually to be able to actually think about how this modified body thing that's part of your own body could become part of you. You are what you eat, but in this symbio future, you eat what you are. And I think this stuff is really important because it hints at documents like this, just released last month, Obama's National Bioeconomy Blueprint. And in these last slides, what I've been trying to talk about is how synthetic biology really affects us as people. And I think that it's documents like this that kind of hint at how our future is going to be controlled by a biology that we can control, that will save humanity, that doesn't necessarily consider what it means to actually experience synthetic biology. And I think that design is a tool that can help us to investigate these issues. A document like this sets out a way that we may want to live, but is it one that we actually have great interest in? And how do we begin to test ideas of what's acceptable and what's not, and also to think up and find new problems that haven't yet been thought of? And I think it was been alluded to a lot today, and part of what I do, the way I approach my work, is I think about it's almost like a science in a way. I use models as useful fictions to test these possible worlds. And I think we need to find a way to do this now before it actually happens. So this was a quick workshop exercise I did with Rachel Armstrong, and it was something I tried to do with a team of iGEM students at Cambridge a couple of years ago. It's like, if you were going to reverse engineer the biotech revolution, what would you make it? What were the ground rules you'll set down? And I think that this is the kind of thing that designers can help with, and artists and other experts. How do we set out what is good synthetic biology? How do we design nature well? So a couple of them I think are really important. Life cycles, not lifespans. If we're dealing with biological stuff, there is no end to the way it lives. It may die, but it becomes part of everything else. And so we need to really focus on these kinds of issues. So this is a draft, and I would welcome any suggestions. And thank you for your patience with the technology. When it's living, it will be very different. Thank you.